Welcome everyone to our 4th annual Society for Reproductive Biology Public Symposium. This session is our outreach session where we open up our conference to the wider world, and we're streaming it online on the SRB website. So each year we focus on one particular aspect of reproductive biology, and we ask some of our leading researchers and leading researchers in our region of our host city to speak. This year, our topic is pluripotency and reprogramming, which are essential properties of stem cells, whether they're from embryos or somatic tissues. We have an excellent panel of speakers, and we're going to have questions in an open question Q&A format at the end. I'm going to quickly introduce the co-chairs of the session. Dr. Deanne Whitworth, waving. She did her PhD at the University of Melbourne with Marilyn Rinfrey and a postdoc with Richard Berenger at MD Anderson in Houston. She then returned to Australia and did a vet degree and worked as a veterinarian for several years on small animals and also horses. And it was when she was in this position that she was seeing stem cell therapies being marketed very heavily as cures for osteoarthritis in dogs and horses. She, in 2012, and she, I think, would admit that she wasn't convinced about the efficacy of these cures. So in 2012, she accepted an academic research position at the University of Queensland in the vet school and has initiated her own research program, which hopefully you heard about in the last session, that's focused on really understanding the properties of induced pluripotent stem cells in domestic animals and Australian native species. Her co-chair is Dr. Hannah Brown. Hannah did her PhD at the University of Adelaide in the lab of Daryl Russell. She then did postdocs with Rex Scaramucci in France and Monica Justice at Baylor College of Medicine in Houston. She returned to Australia in 2012, and her work is currently focused on understanding intergenerational transmission of diabetes. Hannah is also very passionate about science communication. You may have seen her talk about her work on the project, and she's also was able to speak about it to our Prime Minister, Malcolm Turnbull. So thank you. Looking forward to a great session. Thanks to Deanne and Hannah for chairing, and welcome everyone.
Thanks, Becky. So I think you're right. We have an absolutely fantastic lineup of speakers today, and because the talks are going live on the SRB website, I asked all of the speakers if they would tell me something a little personal about themselves. Some of them shared a little, some of them a lot. I'll tell you more later. Yeah, well, I don't know. Is there too much sharing after some of the last talks we saw? Anyway, so the first speaker that we have today is recent-ish to Australia. Professor Nathan Powpan is from the University of Queensland. When I asked him to tell me something about himself, he said that he left the United States to start a lab in Brisbane just before a presidential election. So often he is happy to be misidentified as Canadian. He's going to tell us about his research today about using genomics to elucidate developmental cell lineage decisions. And I think it's going to be a really exciting talk, and I've got some very exciting questions lined up for the end. So thanks, Nathan.
All right, let's see if I can figure out. Go. All right, well, it's a pleasure to be here, and yes, I am newish. I've been here since November, and it's a pleasure to be here to meet some new colleagues and give you a little bit of insight into the work I'm doing. And thanks to the organizers for a chance to be able to present my work. So this session is on big ideas, and I want to present a new perspective on an old idea, and that is how we identify the genetic regulators of cell state. So we can look at this in the context of development. What are the genes that govern pluripotency? Sorry. All right. Well, anyway, What are the genes that are governing pluripotency? What are the genes that govern definitive cell types like cardiomyocytes or endothelial cells? Thanks. And what are the genes that govern the cell transitions through developmental processes? We can look at this also in the context of disease. If a tissue is injured, what are the genes that are governing the response of a cell to that change in the environment? These are fundamental questions about developmental biology and the etiology of disease. Anyway, we have a number of tools at our disposal these days to be able to address these questions. We can look at the nuclear architecture, the interactions within the genome. We can look at the proteins bound across the genome using DNase hypersensitivity. We can look at transcription factor binding or histone modifications that allow us to elucidate specific functions of different parts of the genome. And we can look at the transcriptional profile of a tissue or a cell type. And these give us different pieces of information about what's going on at the level of the nucleus that allow us to be able to dissect some of the mechanisms that are governing cell identity and cell state. Now, I'll say that historically, I'll just make a conjecture here, but I think that there has been, either because of limitations in technology or whatever the case may be, there's been a significant emphasis on transcriptional profiling if you think of quantitative RT-PCR or the array approaches that we had in the past or contemporary approaches in RNA sequencing. The effort to understand the function and identity of a cell has predominantly been focused on understanding the transcriptome. And we've taken, an old approach here that I would say, maybe older, getting older, is to take a population of cells through development or to compare two different cell types and getting this average signal that you might say we have a novel gene that we're interested in and how does it compare to the transcriptional profile of other known genes. And then we can use immunohistochemistry or FACS analysis to look at the single cell level, more specific mechanisms that are related to the expression of that particular gene of interest. But this has gotten us into a new phase of our understanding of transcriptional profiling at the single cell level. So now, instead of looking at populations of cells and taking the mean or the average signal of a particular gene of interest, we can now look at the transcriptome of every single cell, where we're looking at endothelial development here, for example, where we have nascent mesoderm transitioning through to endothelium and the blood lineages here. And every single cell basically acts as an entity in itself, where we can look at the transcriptional profile of every one of those cells and then look at a gene of interest, such as HOPX, for example, and see its distribution within those populations. And this is fantastic. It gives us an excellent opportunity to be able to get at mechanistic insights at a single cell level. Anyway, so these are really excellent technological advances that we have at our disposal these days. But let me just say that there are some significant limitations to transcriptional profiling that we're really running up against some roadblocks in this. And that is that it is only profiling the most highly abundant transcripts. And that's a problem because transcription factors, for example, or other key regulators of a cell's identity that are governing that identity are in very low abundance. And so you're missing out on a great deal of information simply because you can't actually detect it. On the other hand, we're also looking at perhaps differential expression. This is a classic approach where you're looking at two populations of cells and looking at differential expression. And again, this is limited on the abundance. You pull this twofold differential expression, and that's some arbitrary line that says that those that are in highest abundance are somehow more functionally important. But I would say, anyway, what I'm trying to get at here today is that this is actually a major limitation, that we are not actually able to elucidate the fundamental genes that are governing the identity of a cell using transcriptional profiling alone. So I want to back up a little bit here and focus in on some of our newer approaches here, and that is specifically focusing in on ChIP-seq and chromatin dynamics. And so we have an opportunity to be able to identify and analyze the entire genome looking at enhancers, active or primed enhancers, super enhancers, open chromatin, closed chromatin. The list goes on. We have these histone tails that are modified in specific ways that allow us to be able to dissect out specific regions of the genome that allow us then to get at some more mechanism. And I've taken an approach in recent years to try and incorporate chromatin dynamics in a manner that allows us to be able to get more specifically at the genes governing cell identity. So if you have these reads, for example, we'll have a number of different populations of interest over here. We have a number of chromatin marks here as it lines up with RNA sequencing profiling here. And the question that is at hand here that I want to ask is, how do you tell the difference between a structure function gene here, like troponin I, or a transcription factor like NKX2.5 or tau1? How do you actually get at identifying these specific genes, specifically new regulators of cell identity or cell fate, using chromatin dynamics in combination with gene expression? And so I'm not going to go into this. It's very complicated, and honestly, it's very laborious algorithm. But the basic essence of what we've done here is we've taken two approaches. On the one hand, we take our traditional approach of just taking transcriptional profiling. We have a couple of populations of interest, and we look at differential expression. So that's panel number one. That's our one approach. The other approach then is to say we're going to incorporate the transcriptional profiling in combination with chromatin dynamics to see if we can more specifically enrich for the genes that are governing cell identity. And so if we look at the genes that are enriched simply on the basis of gene expression, and this is looking at the cardiac lineage because that's the lineage I'm most interested in, you'll see that by looking at just gene expression differences, we're picking up sarcomere organization and muscle contraction, structural constituents of muscle. And these are the genes that are in high abundance, the transcripts that are in high abundance, like myosin or troponin. These are just functional elements of a cardiomyocyte. But those are not the genes that are governing the identity of that cell. So when we incorporate the chromatin dynamics, the profiles change here. We now are looking at developmental processes, morphogenesis, cell fate commitment, DNA binding. So now we've whittled away all of these high abundance transcripts, and by incorporating chromatin dynamics, now we have a more specifically enriched population of genes that we think are governing the identity of that cell. So let me give you an example of one gene that we picked up using this analysis. And I highlight this particular example because it's not a transcription factor. We have sort of this unreasonable idea that transcription factors are the genes that are governing cell identity. But the cell has a very different interpretation of what that means. And the chromatin dynamics actually create a particular signature of genes governing cell identity, like transcription factors, but not specifically transcription factors. And this is a transmembrane protein that has chromatin dynamics that recapitulate exactly what you might see with a transcription factor. So let me walk you through this. If we have here pluripotency, and the top is H3K27 trimethylation, which is a closed repressed chromatin, H3K4 trimethylation, which is open chromatin, and then H3K36 trimethylation, which is a surrogate for RNA polymerase activity. So in the pluripotent state, this gene, TMEM88, is heavily repressed. And as the cells transition through mesoderm, you say that the repressive mark is still present. And then as the cells transit toward the cardiac progenitor cell, this is replaced with open chromatin marks. So now the repressive brakes are released, and the gene is now actively expressed. And you can see that as it transits on toward a more definitive cardiomyocyte, anyway, that signature dies out. But we carried out functional assays on this, and we found that when we knock down TMEM88, we lose the ability to be able to make cardiomyocytes. This can be rescued with a Wnt inhibitor, which is in keeping with the fact that TMEM88 is an inhibitor of Wnt signaling. And the cells transited instead into an endothelial fate based on this immunohistochemistry for von Willebrand factor. So the key point that I want to make here is that the ability to draw in chromatin dynamics enabled us to identify this novel transmembrane protein that is critical for being able to identify and mechanisms underlying cell fate commitment in cardiovascular development. So in closing, I just want to reiterate my key point here, and that is that transcriptional profiling is good. It's fantastic. It's important. Single-cell RNA sequencing is really amazing, and there are undoubtedly going to be fantastic discoveries made through this, but there are limitations to transcriptional profiling that in terms of being able to get at the key genes that are governing cell identity. And I think that we need to draw our focus and attention toward chromatin dynamics as an approach, a unique opportunity to be able to take the cell's interpretation of key genes that are governing cell identity at the level of histone modifications and this relationship between open and closed chromatin as it relates to gene expression. So with that, I will close. I have a small lab right now up in Brisbane, 2 folks growing, as well as some fantastic collaborators from the United States where I came from last year, and funding sources for my work. So anyway, thank you for your attention, and I think we'll have questions at our panel discussion. Thank you very much.
Great, Nathan. Thank you so much. Okay, our next speaker comes to us from the University of Adelaide. The next speaker is Associate Professor Mark Noddle, and he was the oversharer. When I asked, he gave me 10 facts, and then he edited them and gave me more. But most importantly, he told me that it's his wife's birthday today, that he's missed half of these in the last 20 years. So I did consider that we should sing for her, given it's live, but we're not going to do that. We might do it, after the student dinner, perhaps tomorrow night. But what he did tell me is that his group were the first to produce pigs from frozen embryos. So he's been working in this field for a pretty long time. He's a genuine expert, and he's going to tell us a story of embryonic stem cells, a tale of mice and humans, as well as pigs. Thanks, Mark.
Thank you, Hannah. Thank you for the invitation to speak about some of our work today. And it's my wife's birthday tomorrow, actually, so I have to ring her. Today I want to talk to you about some work we've been doing, and I want to take you back to the beginning about embryonic stem cells. And I want to give you a spoiler alert by telling you now that mouse embryonic stem cells and human embryonic stem cells are not the same. Okay? And this will become clearer as we work through the talk. I also want to finish by talking about some of the work we've been doing in the pig in this area. So mouse ES cells, we know they can differentiate to all the cell types found in the body, including the germ cells. In other words, they are pluripotent. And they were first isolated back in 1988, and this slide just is a cartoon which basically shows the procedures. So we're talking about isolating cells now from the epiblast, which forms following the differentiation of the inner cell mass into the epiblast and the hyperblast. The epiblast gives rise to the fetus and the extraembryonic membranes. So in the mouse, this is around day 4.5, just before implantation. Okay? So that's how mouse ES cells were first isolated. Evans and co. were looking for the equivalent in the embryo of carcinoma cells, which they've been playing around with for a while. So how do we know that mouse embryonic stem cells are pluripotent? Well, if we take embryonic stem cells and we inject them back into the developing blastocyst, we can produce a chimera. In other words, a mouse that consists of the 2 cell types. The cells from the inner cell mass proper, or epiblast proper, and the injected cells. And this is a powerful technique which we've used to do gene knockouts to look at gene function over the last 20 years. And if we mate those animals on with a single mutation, we get animals with a double knockout, for example. And if we see some sort of phenotype, we start to understand what the gene does. So a very powerful technology that's been around for a long time. However, people have tried to isolate embryonic stem cells in other species, such as the pig, for example, and have invariably failed. So was there something special about mouse embryonic stem cells, in particular the 129 strain, or is it, are these cells, can we isolate these cells from all species? So human embryonic stem cells were isolated in 1998 by Jamie Thompson's lab in Wisconsin. So there was a lot of interest in this because here for the first time was the possibility or potential for an effective treatment for a range of diseases and injuries which we can't cure. And most of you in the audience would know these examples. We typically talk about Parkinson's, type 1 diabetes, heart muscle, and also injuries to the spinal cord. And the company who produced this slide, called Geron, was the first company to go to have a clinical trial in the area of spinal injury. Now, I gave the spoiler alert that mouse embryonic stem cells and human embryonic stem cells are not the same. If we look at the mouse, then basically embryonic stem cells are isolated back here. Whoops. prior to implantation. Post-implantation, we see a group of cells called epiblast stem cells. And a number of groups now have shown that human ES cells approximate to these cells in the mouse that are found in the developing embryo post-implantation. So typically we talk about mouse embryonic stem cells as being naive, and we talk about human ES cells being primed because they approximate to the mouse epiblast. So what's the difference? Well, I've told you before that mouse embryonic stem cells can make chimeras if we inject them back into the developing blastocyst. If we take mouse epiblast stem cells, which approximate to the human ES cell type, they don't make chimeras. So based on what I've told you, you can conclude that they're not as pluripotent. So what are the differences? Well, we've talked about naive cells being more pluripotent because in the mouse, the equivalent does not make chimeras. They seem to be more, naive embryonic stem cells seem to be more homogeneous. And this is a comparison now in humans, sorry, I should have mentioned before. More characteristically stable, more proliferative, better replicate embryo development when we make teratomas, for example, with existing human ES cells, the primed human ES cells are out there. What we tend to see is an overrepresentation of the mesoderm germ line. But the caveat here is that we've only had, up until recently, one naive human ES cell to play with, and this was work done by Carol Ware back in 2014. In contrast, if you go on the NIH embryonic stem cell registry, you'll find over 300 primed human ES cell lines which we can play with. So, in terms of their advantages or disadvantages, you'd have to say that the jury is still out in terms of naive versus primed human ES cells. Nevertheless, there are products out there, and the two that I'll mention today, VieCell, a company in the US, have a product in phase 1-2 trials for type 1 diabetes, and here they've taken ES cells, differentiated in part way to insulin-producing cells or beta cells, and wrapped it up in this encapsulation device, and it's now in safety trials in the US. The other one you might have heard of is companies like ACT are looking at macular degeneration, and here they're differentiating human ES cells to retinal epithelial cells which sit at the back of the eye which process light information. And the advantage here is that the eye is an immune-privileged site, so when you start putting cells back into people, they don't tend not to be rejected if you target the eye. So I've talked now about naive human ES cells and primed human ES cells. Now I want to take you back further in development to a stage in embryo development shown here for the mouse, where the inner cell mass is yet to differentiate into the epiblast and hypoblast. So mouse ES cells were isolated from the epiblast. The human equivalent seems to sit further down here, post-implantation, and similar to mouse epiblast stem cells. Now, for various reasons, we looked at the pig. We went back and thought we'd like to try and isolate ES cells in pigs. Other people had tried, so we basically went back to the black drawing board and started again. And I had some very smart people in my lab, and they said, well, we know these cells are more pluripotent than these cells, so let's try and isolate ES cells from this stage in embryo development in the pig. That's basically what they did. To cut a long story short, we used in vitro produced embryos. We get the ovaries from the abattoir, mature the oocytes in vitro, fertilise them and grow them up to the blastocyst stage. So we take these whole embryos, we squash them down on the mouse feeder layers, basically because we don't want the inner cell mass to differentiate any further into the epiblast and hypoblast. And we use serum replacement instead of fetal calf because fetal calf serum tends to promote differentiation. That's not what we wanted. We all did this under low oxygen. And what we ended up with was a homogeneous outgrowth of pluripotent cells, shown here. So we didn't end up typically with a mixture of cells like you do in mice, where you choose ES cells and then just passage them to clean them up to give you a homogeneous culture. Basically, the trophectoderm cells degenerated, leaving us with the inner cell mast cells which became ES cells. If we wait until the inner cell mass differentiates. We end up with a mixture of cells just like you do in mice. So basically what we were doing there, we're isolating these cells at an earlier stage in development than have been used in the past in the mouse. So these cells behave like ES cells. They express OCT4 and NOG, and interestingly they express SSEA1, stage-specific embryo antigen 1, which is now seen as a marker for naive ES cells, as opposed to prime. They maintain their pluripotency. This immunohistochemistry was done at passage 10. They maintain their karyotype. They can be vitrified repeatedly, and they really differentiate to cell types representative of all three germ layers. And we've also isolated this same cell type now in mice and cattle. And we've differentiated these cells, like I said, to other cell types. And this just shows the pig equivalent of retinal epithelial cells, which I talked to you about before, being used, currently trialled for macular degeneration in humans, which is that picture there. So, do these cells make chimeras? Because at the end of the day, this is the gold standard in terms of demonstrating pluripotency. Well, to cut a long story short, we produced chimeric pigs, and we were surprised to see this, but you can see some mottling along the back here because one of the boar that we actually used to fertilise the embryos we isolated our ear cells from was a Hampshire, which has a black coat colour. Now, white is dominant in pigs, so we didn't expect to see this mottling coming through because these cells were then injected to what essentially were white embryos. We haven't demonstrated germline transmission yet because that cell line was female, and female cell lines tend not to go germline, but I don't have time to talk to you about that. So what does all this mean? Well, we believe we've isolated a pluripotent cell type earlier in embryo development that's been compared with those used previously. We don't know if it's the same as a naive mouse ES cell. We're still yet to characterise these cells fully. Do they have added advantages for cell-based therapies? Well, I talked to you before about naive versus primed, and that naive are probably better when it comes to doing cell therapies, although the jury is still out on that one. It would be nice to isolate this cell type in humans, and we're looking to do that if we get some funding. But what about pig ES cells? Do they have a place in this story as well? Well, I think it's one thing we don't have at the moment is a large animal model for human ES cell studies. We have lots of mice ES cells from permissive and non-permissive strains, but we don't have a large animal model. So with the ability to make lots of embryos in the lab, we can look at basic questions about oxygen tension, for example. How easy is it to isolate ES cell lines under 5% versus 20% oxygen, for example. And we have this applied facility, if you like, where we can start to do the sorts of cell-based therapies in a large animal model for the first time. And there are plenty of pig models out there for things like blindness and vascular disease and so forth. So what I wanted to tell you today is basically that mouse ES cells are not the same as human ES cells, and that we believe that pluripotency is lost across development as the embryo develops. We believe the further back you go, the more pluripotent, perhaps even totipotent the ES cell. And I just leave you with a thought by Carol Ware, who isolated one of the first naive human ES cell lines now a couple of years ago. In a review, she wrote that she believed that the full potential of ES cell-based therapies may only be realised if naive ES cells or perhaps something similar are used. Thank you very much.
Great, thank you so much, Mark. Our next speaker comes to us from the University of Melbourne and is Professor Christine Wells. Christine told me that she wasn't funny and didn't have any funny stories to tell me, so I Googled her. Turns out she might not have any funny stories, but what she does have is more than 1,000 followers on Twitter. So, which I think is incredibly impressive and something I aim for in life as an avid science communicator. So you can follow her at Mincle, M-I-N-C-L-E. And you can see what she's up to when she's not doing science in the lab, although she does Twitter a lot about science. So not only is she doing extraordinary science, looking at she describes stem cells as a pocket watch for changes. An ideal cell type. I can't speak. To look at change. So when she's not communicating science, that's what she's up to. Thanks, Christine.
Thanks. Thanks to the organisers for the invitation to come and speak. I've really enjoyed the meeting so far. So we've had a great introduction to stem cells and pluripotency already, and we've also had a lovely introduction, thanks to Nathan, on transcripts and transcriptional profiling. And I wanted to open my talk by just reminding you that tissues and cells can be grouped or clustered by the networks of genes that they express. So Nathan's quite right, transcriptomics is an old method. It's still a workhorse because it is the single technology where we can gather the most information from a single population in one hit. And we can't do that, unfortunately, at the moment for proteins, and we certainly can't do it for metabolites. And what you're seeing here is simply every dot represents a different cell or a different tissue type in an atlas. This is the Phenom 5 atlas, which was published a few years ago now. And the second point I want to make from this slide is the fact that when you profile cells in isolation, it's very difficult to understand what the networks of genes that they're using might mean, because you're capturing probably around 12,000 transcripts for any group of cells. But when you have an atlas approach and you can compare large numbers of tissues against one another, very quickly you are able to see which types of transcripts are able to group cells with similar anatomical or developmental properties together. And patterns that may be anecdotal in an individual experiment become generalisable and even rules when you start looking them en masse like this. So these are incredibly valuable resources. The difficulty with transcriptional profiling, and again as Nathan's indicated, is that it's just one part of a layer of molecular information that contributes to how we describe the phenotype of a cell. DNA here is the easiest part of the information to gather because it's relatively invariant from cell to cell and actually relatively invariant from person to person and actually relatively invariant from species to species, although there are clearly some caveats to that last point. The difficulty here is that we think we've got between 20 and 30,000 genes, but we actually only know what about 7,000 of these genes do. 7,000 genes which we've annotated to a pathway or a network reliably. And for the rest, we might have a name. That name might be associated with some kind of protein domain. It may be named after a tissue it's been discovered in, but we know very little about their functions. And an even greater proportion of protein-coding genes, an even greater proportion of genes are not protein-coding, I should say, but they are non-coding RNAs. And their role probably extends across this entire molecular network in terms of not just designating phenotype, but also stabilising that phenotype as well. Of course, as we move into those molecules which are produced in response to environmental stimuli, RNA, protein modifications, and of course the entire metabolite protein and metabolite genome interaction, then the level of complexity that we're dealing with also increases. And this is a reminder to us that cells are not static, that we can't take one individual RNA snapshot and really understand what the phenotype of that cell is. We need a way of integrating all of this information together and filtering it down to that subset of really relevant molecules that can tell us something about the phenotype that we're interested in. And then, of course, we need to remember that there is a level of variability in biological systems that's not just a reflection of a different way two people in a lab might pipette, or different genetic backgrounds that we all contribute to these kinds of biologies, but actually also represent a fundamental flux, a fundamental component of cell systems themselves, because cells are responding to environmental cues and then returning to a stable state, because cells are dividing. We can think about these networks of genes and proteins and other molecules constraining the phenotype of a cell such that some cells are going to be deep down in this phenotypic well. The entropy for change is going to be very, very low. They're going to be resistant to moving from this state. Perhaps they don't express the receptors of the growth factors that are being challenged across them. Perhaps they're just about to enter, or they've just moving through the cell cycle, so most of their energy is going into cell division rather than differentiation. And in this stem cell population, we have just a very few number of cells which are not in the cell cycle G2 or M phases, but are expressing growth factor receptors, and where entropy for change is high, but our understanding of which way they're going to change is low. So networks don't just enable the phenotype, they also constrain the phenotype of a cell, and they buffer the phenotype of a cell from these extrinsic events. And I think some of the questions that we should be asking of these stem cells is really, how stable is the phenotype that we're measuring? How predictable is any direction of change as we differentiate a population of cells? And more importantly, what is the flux in the system, and how do we use this to our advantage? If we took this network, this pluripotency network, and compared human, mouse and pig, we know we're going to see differences. Some of these are going to be because the cells may not be equivalent states. Some of them may be because different species simply use different networks. And Dee showed us this very beautifully last session. So we really need to understand that variability in this network is a pretty important factor, and we should be exploring it. This picture of Waddington's epigenetic landscape is in most biological textbooks, and I think most of you would be familiar with it. And if any of you are playing bingo, I guess now would be the time to hit one of the bingo buttons. But I often think that this particular diagram is misunderstood. And I really like Conrad Waddington's description of this landscape. So what he's describing is the canalisation of a cell through development. And originally he talked about it in terms of these bifurcations or decision points, at which there was really a point of no return. A cell moved down this landscape, it funneled down into one of the ruts, and then you ended up with a differentiated cell type. And we consider that the poles underpinning this landscape are the networks of genes, which might be describing the pluripotency network here, or the differentiated cell network here, and that these humps are really describing the epigenetic barriers which prevent a cell moving between these different regions. But of course, Waddington also talked about canalisation as allowing variability. In a canalised system, a cell can actually take a different route down any one of these valleys. What you're looking at is the path, the common path. Oh, that was good. What you're looking at here is the common path, but of course a cell could run down the edge and still reach the end here. So canalisation allows for a level of phenotypic variation, and I think this is important when we're thinking about expressivity of phenotypes, penetrance of genetic mutation, and when we're talking about including variability in our networks. So last year, the Phenom Group, which published the big transcriptional atlas, also looked at differentiation in a large number of cell types. Here across these x-axes you can see time measured in minutes, and they took a series of different cell types, including quite a large number of pluripotent cell types, and differentiated down different mesendoderm and neuroectoderm lineages. And they asked, what are the classes of transcriptional change that you see? And what you're seeing here is that the very, very first thing that happens, really within the first few hours of change, the first hour indeed, is a change in the enhancer landscape of the transcriptome. And then you see a change in the transcription factor genes, and then you see a change in the structural genes. So transition out of a pluripotency network requires first remodelling of the chromatin landscape, and then instruction of the new transcription factors for the next canalised network, and then the structural proteins, which are going to be the proteins which actually do the function of that new cell state. What about reprogramming? If we're taking a cell and we're moving it back up this barrier, and we know, thanks to Yamanaka, that all that is needed to reprogram a cell from a differentiated state back to a pluripotent state is the expression of 3 or 4 transcription factors. That's enough to reprogram. So Andras Nagy, as part of the Project Grandiose team, asked this question in mouse. He took a fibroblast from a mouse that had already been reprogrammed once. It had been reprogrammed with a piggyback transposon, which could be turned on and off with the presence of doxycycline. And when he measured reprogramming under a low level of doxycycline, he was able to recapitulate the normal induced pluripotent cell state, or this normal ES-cell-like state. But when he exposed the cells to really high levels of doxycycline, he got this funny-looking cell. They called it the F class because it's a fuzzy-looking cell. And the fuzzy cells turned out, through the chimera assays, to be pluripotent. But they don't share the same transcriptome as an ES cell, and they don't share the same epigenome. In fact, these are quite synthetic cells. They don't last in the absence of this high dox. So you have to have very, very high levels of NANOG in particular superimposed on these cells to maintain that cell state. So this also gave us some insight into the fact that we are often constrained when we talk about pluripotent stem cells to thinking about those cell types that we know from developmental or anatomical niches. But actually, there are opportunities with these stem cells to be exploring landscapes well outside our what's known. Combinations of networks and genes in networks which haven't been seen before and which can give rise to completely new cell states. And this era of synthetic cell biology, I think, is very exciting for its potential. The other thing that was discovered in this Project Grandiose program was that, and I think I've got a slide for it here, the very first changes that happen as you move a fibroblast back up that Waddington landscape to a pluripotent-like cell is the small RNA and transcriptome changes first. In fact, it changes very rapidly. But the epigenome, and in particular DNA methylation, this is the last thing to change. And so what we've learned from this is that one of the roles of permanent chromatin changes, such as seen with mitotically heritable epigenome changes, is that this stabilises cell phenotype rather than driving cell phenotype. So we often think about the genome as a book or a map, but I'd like you to think about it more as a library. It's a library with almost infinite numbers of stories to be told. And then, if we consider that, then this idea that a stem cell has got every shelf, every possible story available to it doesn't really make sense. In fact, there's a separate stack just for the pluripotent stem cells, and different pluripotent stem cell types might be reading a different storybook. And to get this stem cell through to a differentiated cell type, it actually has to transition through different cell states. It's very hard to get it to directly go to a mature cells. And these epigenetic factors, like librarians, their roles are to organise the stacks and just to bring the books that the cell needs to the front of the library. So their role is to stabilise, not to direct state change. In the very last part, I want to point out to you that moving through all of these layers of data can be very confusing, and for non-bioinformaticians or people who, unlike me, don't like looking at sequence, it can be very hard to know how to get traction onto these. So systems biology has got some fairly rigorous principles to move through different types of data. You've got this idea of emergence, which is the idea that combining everything, you get something bigger in return inside. Modularity, this idea that proteins work in networks and pathways. And this idea of robustness, which means that you can have a level of phenotypic variation while still belonging to the same phenotypic class. And so in order to navigate this, we need not just to generate lots of good quality data, we need tools for looking at it. And I really just want to throw at you a few screenshots of this platform that we've built called Fanatics. It's a free resource, it's available to all of you. It's got lots and lots of interesting information in early embryogenesis, from primary embryogenesis and directed stem cell differentiation. It's easy to search, Google-like terms. It's got ways of comparing the expression of different genes. Here's beta-actin, it's expressed pretty ubiquitously. Here's OCT4, it's really expressed a whole lot in pluripotent cells and not in anything else. Here is DMT3L, which is really restricted to very early embryogenesis, the early blastocyst stage in humans here. You can look for your favourite gene. In this case, I'm looking for Minkle, which is my favourite gene, and you can see that your gene may be expressed very highly in a certain cell type. Here, Minkle's really highly expressed in innate immune cells and not at all pluripotent cells. And you can dig in and have a look at the individual data sets that have contributed to this profile. So it's really handy if you just want to know whether the gene you're interested in is really that unique to a particular cell state, or whether it's reproducibly seen in the cell state you're interested in. You can build heat maps if you've got gene lists that you're interested in looking at. And that's it. Really just to acknowledge that I talked about the Phenom work, which is a very large program run by RIKA in Japan under the guidance of Yoshihide Hayashizaki, Piero Konichi and Al Forrest. And I talked about Project Grandiose, which was the brainchild of Andres Nagy in Canada, but had a number of Australian contributors. Thank you.
Great, thank you so much, Christine. Now might be a good time to tell you that if you are on social media, there is a hashtag for the meeting. I would read it to you, but it will take longer than the two next talks. No, that's a joke. It is hashtag ESASRBANZBMS for all the societies meeting 2016. It may well be the longest conference hashtag ever. The next speaker in today's session is Caroline Gargett from the Hudson in Melbourne, and she's going to tell us today about uterine stem cell isolation and functions. But it turns out the stem cells in the uterus aren't the only stem she does, because I heard a story that she may well be one of the women in STEM pioneers, because she started her PhD later in life, but her three sons all avoided science because they thought it was a girl's job. So it seems like she's doing a great job encouraging men away from science, or I shouldn't say that, but promoting females in science and doing fantastic research on uterine stem cells, so thank you.
Yeah, so thank you very much, and it is quite true. My sons were steered away, not by me, that was their interpretation. I'd like to thank the organisers for the opportunity to talk to you today. And the cell type I'm going to talk about is a lot more differentiated than the cells that you've heard about so far. I think they're still in the library, but they are a mesenchymal stem stromal cell, and particularly focus a little bit on the uterus, but some people don't even believe they are a stem cell, and there's good reason why they think that. lot of it's to do really with what's being characterised. So let's just define what a mesenchymal stem cell is, or some people call them a stromal cell. The International Society for Cellular Therapy defined them quite some years ago, about 2006, as clonogenic, plastic adherent, multi-potent cells that differentiate into multiple mesodermal lineages, as shown up here. And they have express a particular surface phenotype. And that's the unfortunate part, because people hang on to that surface phenotype, and that surface phenotype is on fibroblasts as well as many other cell types. So many people are not working with mesenchymal stem cells, or MSC. Now, why are MSC important? They're important because there are many clinical trials currently in place using MSC for cell-based therapies. In fact, there's over 500 that are listed on the NIH website. And over 2,000 people have had these cells in one form or another for a whole raft of disorders. They're mostly used for disorders associated with chronic inflammation, and they actually rely on the non-stem cell property of these cells, their anti-inflammatory and immunomodulatory effects. And in fact, those components are really often due to the fibroblasts that are in those cell populations that are being given to the patients. So where are MSC sourced from? Originally they were sourced from bone marrow. More recently, liposuction and sourcing them from adipose tissue has become really popular. Both of these are highly invasive techniques, and they require an anaesthetic. And in fact, someone in Australia has died from the liposuction technique. So it's not, entirely safe, even if we've got a lot of fat to get rid of. Reproductive tissues actually offer another source of cells, and these are easily obtainable because some of the tissues, like umbilical cord or the placenta, are waste tissue, and these can be salvaged to obtain these MSC. We focused on the endometrium, the lining of the uterus, of a highly regenerative tissue. These too are very easy to obtain in an office-based procedure, just using a Powpan biopsy like this. And we discovered in human endometrium, but also in mouse, but in human we discovered the MSC using cell cloning as a technique, just a very basic, simple way to find them. We then went on to characterise these cells for, as it were, adult stem cell properties. And we found that they underwent self-renewal, they're highly proliferative, could produce large numbers of cells from a single cell, and they underwent multi-lineage differentiation. And these 4 lineages that I show you here, they were from a single cell derived, okay? So they really are multipotent cells. They expressed that MSC phenotype, which really doesn't mean terribly much, but and we made the conclusion in the end that human endometrium contained a fairly rare population of cells with MSC type properties. But having clonogenic cells to study is not terribly useful, and we really spent some time to try and identify markers for these cells so that we could actually study them properly and perhaps exploit them for clinical use. Our first set was 2 markers, co-expression of CD146 and PDGF receptor beta, and this isolated a small population of cells. They happened to be perivascular cells, or pericytes, which is the value of identifying markers. And we then went on to screen for a number of perivascular markers to find a single marker called SUSD2, so that we could pull out these cells. This just shows where we could identify the cells. There's a pericyte here. The SUSD2 is more of a perivascular cell, and we could find them also in postmenopausal endometrium, and they don't express the estrogen receptor alpha. The important thing was the location of these cells in the endometrium. They were around the blood vessels in the basalis layer, which is not shed at menstruation, where we hypothesised they would be. But they were also up here in the functional layer, and this is what's shed during menstruation, but it's also available when you take an endometrial biopsy, in your office-based procedure. So it's a much... we knew then we could obtain these cells very readily for use, potential use in cell-based therapies. And we did all the tests, the ISCT tests for their MSC properties, but we also were able to reconstitute stromal tissue in vivo. Now, I just like to go through some of the impediments and some of the issues that have arisen, because really what happened with the MSC field is things jumped into the clinic far ahead of the science, and how all the issues are now being raised because they haven't really been as efficacious as one had hoped they might have been based on the kind of properties that they have in vitro. And most of the problem is that they're lacking quite a bit of efficacy in the clinical trials. Much of this is due to this heterogeneity of cellular product. They're really not purified to begin with, and often it's just fibroblasts. And this just shows you, say, fat off that liposuction. You know, it's sort of, as it were, kind of blended a bit and then spun down and given back to the patient. What's given back is just the cell pellet, and it's uncharacterised, and there's a large number of different types of cells in there, besides the perivascular MSC. The other thing that happens if you do isolate the perivascular cell that's clonogenic and has the high proliferative potential is that if you grow them up in culture, which you need to do to get the cell numbers, they'll spontaneously differentiate. And so then you're giving back, again, a small lot of MSC with a large number of fibroblasts. And there's been a very nice study done by Linda Gadish, who's done just a microarray profiling to show that this spontaneous differentiation from the fresh to the cultured cells using a set of signature genes for the MSC and then for the fibroblasts, and she showed that very quickly in culture the MSC, based on our mark as a CD146 PDGF receptor beta population, became like the fibroblasts quite quickly, the CD146 negative PDGF receptor beta positive cells. And the other reason I think why these cells haven't perhaps been as useful clinically is because they're given by intravenous injection. And the thing is, if you give a lot of cells, there's millions, say 70 million cells, if you give them by intravenous injection, they're going to just get trapped in the lungs. It's an incredible waste of dose. 90% are trapped in the lungs. It's all right if that's what you're treating. And they are attracted to the site of injury, but they really disappear well within 48 hours. They do have effects. They have a systemic effect. I think there's a lot of cytokines produced, but all the same, it seems an incredible waste of dose. Then there's the concept of which way do you go, and the business model is allogeneic is really preferable. I happen to favour autologous, which is not a very good business model at all, but if you want to give repeated doses, and even though these cells are lowly immunogenic, they still are immunogenic, an autologous approach to me seems a much better way to go. And really, the mechanism of action is not really that well known because, again, the work jumped into the clinic far too quickly ahead of the science. So we're working towards clinical translation of our endometrial MSC, and we're looking at an autologous approach. And we're trying to treat a disorder, which I'm not going to talk about at all today, but it's incredibly common but hidden. It's a women's health issue, and it's called pelvic organ prolapse. And what we're trying to do is culture up our cells. And we've got now our single marker and a very simple way to isolate the cells with minimal damage. We've been working towards culture expanding these cells using good manufacturing practice guidelines. And here we now have installed and commissioned, it's going to be commissioned next week, at our institute, the Hudson, a biospheric cell isolator unit. And so this is in place of a big, bulky and really hard to access clean room where you can move around the outside here and grow your cells, isolate and grow your cells in whatever condition you like. And where we've got to with our cells so far, where you have to get rid of all the animal products, we're down to serum free. We're not got rid of all the animal products yet. We've got the serum gone. We find that they grow much better in 5% oxygen and on the fibronectin, and we can generate large numbers of cells quite easily. But the thing is, what we're trying to do now to deal with the problem of the spontaneous differentiation is to block that during the culture expansion process. And we were looking at small molecules to inhibit that differentiation process. And we tried a bit of a candidate approach, and actually we borrowed a lot from the embryonic stem cell literature. And we've been using A8301, and I noticed someone else had that. It was Nathan in one of his culture systems, maybe Mark. But it's a TGF-beta receptor inhibitor, and if you're going to use autologous, you really have to have a standard for your product that's the same and really difficult for every different patient. But this is what we were trying to do, and what we found with the A8301 is it did promote proliferation. You can sort of see this here, just in these microscope views. It blocks, the main effect it has is it blocks apoptosis and senescence, actually. We do get many more clonogenic cells as a result of that. And we find we can monitor our cultures now using this SUS-D2. And what we've found here is that when we've treated our cells with the A8301, and these are late-term cultures, and the effect is greater in the older MSC from older women, is that we can bring them up to around about 90 to 95% all the time. So it's for most patients. So this is really quite useful from a quality assurance point of view. We then wondered whether this molecule would be really quite good for all the other MSC types, and so we've been investigating that. And you can see this is very much a work in progress, and we haven't got very many N numbers for some of the ones we've tried. But our hatched bars are where we've treated with A8301, and you can see in all the endometrial ones we get really quite high. And in the placental ones it's improved. And in the bone marrow, which are really quite difficult cells to grow, we make a big difference, but that is only N equals 1 when we're working on it, and we're currently working on the fat as well. Now, one of the other things that we regard as really important before we go into the clinic is to test all these things in vivo. And so Shanti Gurung, who's been doing this work, a PhD student, so just looking at our A8301 treated cells, will they be good and will they be okay after that treatment when we put them into the animal? And so she's just labelled them up with a M-cherry using a lentivirus to track them, put them under the kidney capsule, and this is about 14 days later. You can still see a number of cells there, although it is diminished. And she's been checking with ALU sequences as well, and K's kidney and S's skin, and they're pretty okay. They're at least as much, if not more, than the control cells. So it's still really a work in progress. But the other areas where we're trying animal models is before we go into the clinic with our pelvic organ prolapse surgery, using a tissue engineering sort of approach, where we put our EMSC on a mesh and gelatin composite to implant into women. We've been testing them first in a small animal model, and here we've done a fascial wound defect. This is a xenogeneic model, so human cells into rat. Now, the cells lasted about 14 days, a little bit longer than an intravenous because it's local delivery, but still not that long. But they did have quite a dramatic effect. They stimulated an early neovascularisation. They promoted quite a strong inflammatory response initially, but that changed to the M1 inflammatory cells to an M2 wound healing type after about 30 days, even though the cells were already gone. And overall, the biocompatibility was markedly increased, the much lower chronic inflammatory responses is at 90 days. But I think one of the main things that really happened that was really different was the kind of collagen that was laid down. You can see this crimp here versus the scarring type collagen without the cells. This crimp gives the biomechanical properties that are really needed. And so what we found here, and this is some biomechanical kind of testing, but really what you want to see is less stiffness, and this is the slope. So the slope is lower, and a longer period here, and that's kind of the distensibility, and this happens because you've got crimp and it can be extended. So we were quite surprised at this effect, which is actually only a paracrine effect because the cells haven't really survived that long. But before we go into women, this is vaginal repair surgery from prolapse, birth-induced injury, we want to test our model truly as we will do in women. So we've gone to the ovine model. Sheep vagina is very similar to the women. Here now we're taking the sheep endometrial MSC, putting them on the mesh and implanting them vaginally using urogynecologists to do the surgery. But of course, our SUSD2 didn't work. It's not expressed, or the antibody doesn't react with it. But we have used another bone marrow MSC marker, CD271, to get out some perivascular cells. The perivascular cells are in a slightly different position. They're in the adventitia of these vessels. And this is known in other systems, that the adventitial cells, perivascular cells, are also just have the MSC type properties. We've done, this is a very small number, but very encouraging experiment where we put the ovine EMSC, cultured them up, put them on the mesh, inserted it vaginally. This is 21 days later. We had to harvest at that point because this had to go into a grant. But you can see, and you can see we labelled them with an antivirus at this point, and you can see large numbers of cells in a sheet, all looking very viable. So we're feeling that this is really quite encouraging. So this is using autologous cells and local delivery. So just to summarise, in a human endometrium, we have a source of cells from a reproductive tissue that it really is minimally invasive to obtain. It's a highly regenerative tissue, and we think that the cells that we've been isolating are actually involved in the vascular stromal regeneration that occurs every month in these in women. We are developing simple protocols to isolate the cells, and certainly the human cells are much easier to isolate than the sheep with the SUSD2. And we're addressing the issue, some of the issues that are facing the MSC field in that spontaneous differentiation in culture by using this A8301. And we're looking really at other molecules too in more of a screening approach. But what that really does is to produce a reproducible product for autologous use, which is quite important when you're having to get something similar or the same from every single patient. And we're trying to determine the mechanism of action of these cells in preclinical models, and we've shown in a xenogeneic model that they have a paracrine action. But in a autologous model, and modelling exactly what we want to do in women, there is the potential, it's too early to say, that maybe there is some cellular integration because we've addressed all the kind of issues that need to be addressed. And finally, I would like to acknowledge many of the people in my team, both past and present, who've contributed to the work I described to you today. And in particular, I would like to acknowledge our clinicians, who Anna and Nathania have shown here, who have helped us hugely along the way with advice and with the surgeries. This is James and Shanti, whose work I described quite a bit. But I've done a lot of work with CSIRO, who've designed all the material side of it for us, and they're tissue engineers as well, and also all the funding bodies. So thank you.
Thank you so much, Caroline. Okay, so our final speaker for today's session is Patrick Weston, who is also from the Hudson in Melbourne. Perhaps like a naive stem cell, Patrick was naive and didn't give me any information about him, so I thought I would Google. But the good news is, Patrick, we're running a little late, so I'm going to give one little ditto. It turns out Patrick has been in the field of reproduction for quite a while, but has not always worked in stem cells. I found that he worked, to begin with, in temperature-dependent sex determination in American alligators. So obviously he has a quite diverse story, and hopefully he's going to tell us today about drugs, germline epigenetics and offspring health. Should we be concerned? Thanks, Patrick.
Okay, thanks, Hannah. So I'm going to move on to a different type of stem cell, and these are the germ cells in the body, which are transmitted via oocyte and sperm to the following generation. And of course, these cells are ultimate stem cells in that they have to make a whole new individual every generation, so they're totipotent. I particularly want to focus on epigenetics and the role of germ cells in reprogramming and whether or not epigenetic information in germ cells changes in response to particular environmental stimuli, and in this case I'm going to use the example of drugs. So I guess this audience needs no introduction for sperm and oocytes and their role in transmitting genetic information to the offspring, and this genetic information forms a template, of course, for the whole of life in the offspring. However, it's perhaps not as widely known that germ cells also transmit epigenetic information from the parent to the offspring, and this information is critical for proper development of the offspring. This is particularly important to understand, I think, because epigenetic mechanisms are responsive to environmental cues, and particular environmental stimuli can change epigenetic state in cells. And so whether or not these environmental stimuli can change outcomes in the germline, leading to differences in the offspring, is essentially what this talk is about. And it's a topic that I think is particularly important in understanding both evolution and also disease in humans. So there are quite a number of studies now which indicate that the environment of the parent can change outcomes in the offspring. For example, if you take male rats or mice and you feed them high-fat diet, then the offspring of genetically identical mice differs depending on the diet that you've fed the father. And so this indicates that the environment of the parent, or particularly the father in this case, can change outcomes in the offspring, and it's broadly thought that this is due to epigenetic differences in the germlines that are transmitted, or germ cells that are transmitted. So what is epigenetics? Now, I don't really need to introduce it, I guess, now, because we've covered this topic in the last few talks. However, it's been stated a couple of times today that epigenetic mechanisms essentially stabilise lineage identity of cells, or the functional identity of cells, and this is achieved through broad regulation of transcriptional profiles in those cells, through switching off genes and switching on genes in a stable way. So if you repress cohorts of genes or you allow other cohorts to be activated, and of course every cell in the body contains the same DNA but has many different outcomes in cellular phenotype, and this epigenetic mechanisms are largely responsible for those phenotypic differences. So the best described epigenetic modification, I guess, is DNA methylation, which we've known about for some time now, and this is a change directly to the DNA, a chemical modification to cytosine in the DNA sequence, but doesn't change the DNA sequence itself. Other epigenetic modifications include histone modifications, and these are chemical changes basically to the histone proteins around which the DNA is wrapped, and this then allows the DNA to be packaged in either a more tightly or in a more relaxed form, so that genes can be either repressed in tightly packaged chromatin or accessible in open chromatin. And so this allows then repression or potential activation of whole cohorts of genes. So the germline plays a particularly important role in controlling epigenetic information. Early germ cells form during embryonic development, and they form, this is essentially a cartoon for mouse, but they form a gastrulation from a number of cells which would otherwise make somatic cells. So the germ cells are set aside, and at this stage they contain epigenetic information which is essentially equivalent to their surrounding cells. So they then undergo a process of epigenetic reprogramming, and this removes epigenetic information that is in these progenitor cells and results in a cell that's essentially naive for epigenetic modifications. It essentially has a clean slate on which new epigenetic information can be established. So this allows then either male programming or female programming in the germline during spermatogenesis or during oogenesis, and the establishment of an epigenome in sperm and oocytes which is not equivalent, which is different between the sexes, and at fertilisation these differences come together to complement and allow the functional expression of the male and female genomes and the formation of an embryo and proper development thereafter. So our work really is focused on a histone modifying complex known as polycomb repressive complex 2, PRC2, and this complex is made up of 3 critical proteins or core proteins, SUS12, enhancer of zest and EED. So I'm going to talk about enhancer of zest and EED today. These catalyse an epigenetic modification known as trimethylated K27, which leads to this compressed chromatin format and the repression of cohorts of genes. And many of these genes include transcription factors such as homeobox genes. These changes are mitotically heritable and allow stabilisation of gene expression patterns through time and cell function and identity through time. So we initially wanted to ask whether we could, in the developing germline, whether if you alter polycomb function, whether you would then affect outcomes in the offspring. And this would then indicate that there's a heritable epigenetic process that underpins some of development in the following generation. So I'm going to briefly go through some data, and because this is a public forum, I didn't want to do too much detail. But the first experiment we did was that we had males with mutations in the EED gene, so this is a critical component of PRC2, and we looked for differences in offspring in the following generation using males mated to wild-type females. So the experimental plan is shown here. We have wild-type females. These females are mated to males, which are either homozygous for the mutation or heterozygous for the mutation in EED. So these guys here have sperm which have developed in the absence of any good copies of EED, whereas these guys here have sperm which have developed in the presence of one good copy EED, and we consider these to be relatively normal control. So these males can produce heterozygous offspring in both cases, and so we can compare these heterozygous offspring, and these animals are genetically equivalent for EED, and we look for differences which we then would indicate that the epigenetic state of the sperm coming from the homozygous males is different to those coming from the heterozygous, or at least functionally different. So this is one piece of the data that we have. We did an RNA-seq experiment and a microarray experiment on embryos which we produced from homozygous fathers or heterozygous fathers, and we looked for transcriptional differences in these offspring, and we found a bunch of transcriptional differences which indicate that the outcomes in these offspring are different between the homozygotes and the heterozygotes, and that that polycomb somehow regulates paternal epigenetic inheritance, and so there's at least an effect down the paternal side. So we then wanted to ask whether we could see similar effects on the maternal side, and so we did the same type of experiment, but this time we use a specific model in which we can delete enhancer as S2 or indeed EED specifically in the growing oocyte, and ask whether this then affects outcomes in the offspring. Initially, we confirmed that trimethylated K27 is enriched in growing oocytes, and you can see primordial oocytes over here and growing through at day 2. And day 24 here you can see nice enrichment in a growing oocyte down the bottom. So this is depicted here through the growth of oocytes for H3K27 enrichment, and when we use a model in which we can delete enhancer as S2 in the early growing oocyte, we can see that we can almost completely block the enrichment of K27 in the growing oocyte. So then the question is, if we block this process, does this affect outcomes in the offspring? And so the data we have so far is that offspring from oocytes that lack or are heterozygous for enhancer as S2 are significantly different in birth weight, 25% lighter on the extremes of this, and this indicates that there's a maternal effect, at least in the oocytes. This actually is not a new discovery, and there was a paper published 13 years ago from Sarani's lab, and I was actually in the lab when this work was done, that showed exactly the same thing, so the data wasn't a surprise for us. We've also done this experiment with EED, and if you want to hear a bit more about that, then you should go to Lexi Prokopuk's talk on Tuesday, I think it is, at 11:30. So this indicates that enhancer as S2 is a maternal effect protein, which then affects outcomes in the offspring. This is of particular interest because enhancer as S2, of course, is not only expressed in oocytes, it's expressed probably in all cells at some stage in their development, and this regulates K27 methylation and the genes which are affected by that epigenetic modification. It's also misregulated in many cancers. So enhancer as S2 is becoming quite a popular gene in oncogenesis, and if you look in PubMed and do a search just in 2016 for an enhancer as S2 in cancer, you'll find about more than 220 publications just this year alone. There also has been quite a focus on enhancer as S2 for development of anti-cancer drugs for this very reason. And so these drugs block enhancer as S2 function and offer quite exciting new therapeutic possibilities for patients. I think this is going to become, or obviously is becoming a big area in cancer therapy. One of the targets, or one of the cancer types in which you find excessive amounts of enhancer as S2 is non-Hodgkin's lymphoma, quite a common tumour. Typically affects people in older stages of life, or later stages of life, but also affects a certain number of people of reproductive age. So these patients, some of these patients are now in clinical trial using anti-enhancer as S2 drugs. And it's potential, obviously, that the enhancer as S2 drugs may affect the germline in these individuals of reproductive age. Just these individuals here affect around 12,000 new cases a year in the States, about 1,000 in Australia and 2,500 in the UK. So the question for us then is whether drugs that block enhancer as S2 and prevent the enrichment of K27 might affect the germline in these patients and therefore outcomes in the offspring. And so the first question we asked was in fetal germ cells, and these won't normally be exposed to the drug because you're not going to be pregnant when you take the drugs. But we can see in germ cells at least that we very significantly deplete K27 in the germ cells in a dose-dependent way. So if we look at germ cells a bit later, now we're in oocytes, and these are growing oocytes. These have been treated for 7 days, and you can see a significant reduction, which is quantified here, in K27 over a week of treatment from the essentially birth for a week of treatment using the drug. And I'll just remind you that if we knock out enhancer as S2 early in this process, then we completely deplete K27 in growing oocytes. So I guess the question for us is whether or not these drugs might lead to changes in the offspring that are similar to those that occur in mice. And so we then want to look at the guidelines for using these drugs in the patients, and this is a summary of from one of the studies. Of course, you should not be pregnant when you start the treatment. You should not become pregnant during the treatment, and you should not become pregnant for at least 30 days after treatment. However, oogenesis in humans actually takes quite a lot longer than 30 days, and so obviously after 30 days, some of those oocytes may still see the effects of enhancer as S2 blockage and have epigenetic differences. At this stage, we have no idea whether that's the case or not. And so I think at the moment, there's a case for looking at the guidelines of use of epigenetic modifying drugs, not just those which target enhancer as S2, but other epigenetic modifications as well, and generating guidelines which specifically take into account the possibility that blocking epigenetic mechanisms in the germline directly could affect outcomes in the offspring. And that's essentially where I want to leave it for further discussion if anyone is so inclined. Just like to recognise the people who did the work, particularly Lexi Prokopuk and Jess Stringer and Kirsten Hogg in my lab, who've done essentially all the work in the mouse models and the drug work that we've done so far, and a number of people who supplied mice. Thank you.
Great, thank you, Patrick, and thank you to all our speakers. Maybe we can give them all a round of applause while I invite them up to become a panel. Don't go anywhere. If you have a question, prep it, because we're about to get going on the panel discussion. So let's thank them while they move up here.
So I get to ask the first. So I think this is on. Yes. Actually, can I pose a question to Mark on your pig ESCs? So with the discussion of naive versus primed, one of the key hallmarks of naive, especially if the cells are female, is that both X chromosomes are active. So have you looked at X chromosome status in your cells?
The short answer is no. But that's one, as you said, that's one of the defining characteristics. We're focused actually on demonstrating germline transmission. We think that'll bring the money in.
Okay. Questions? Anyone?
Building on that, Mark, didn't you say something in your talk about that the female cells don't go germline, but you said that there was a reason for that, and what is the reason for that?
That's a good question. We know from the stuff we did a long time ago in mice, and other people have shown this too, that female ES cells tend not, well, about 20% go germline when you make the chimeras, whereas for males it gets closer to 100%. And probably 10 years ago now, we did a similar experiment in pigs where we took inner cell masses from male and female embryos and showed the same thing, that we made lots of male chimeras and 100% went germline, and in the females I think it was closer to 20%. So I'm not sure the reasons for that, to be honest. It depends on the sex of the embryo that you put the cells into too. So there's probably a few PhDs there. So I don't have the answer, sorry.
Could it possibly be though, if you've got cells that are supposed to have two functional X chromosomes and only one is active, that's why they're not going germline? Yeah,
I think if you put 2 and 2 together, and I think you come up with 4. So I tend to agree, but we haven't done that.
Questions?
I've got one while the audience are thinking of one, because I'm sure they've got one in a minute. I guess the common themes across all of the talks were that not all stem cells are the same, that what you capture is perhaps a sneak peek at just a single point in time, and that you need to analyse multi-layers. So if we don't have time to read the whole library, which book should we read? You know, what should we all be working on on something common, or is there, or is the diversity in the 500 cell lines representative of the fact we need to explore everything? And I guess the question can go to anybody.
Well, my perspective is the more varied the models that we have available to us, the more useful the books are that we have available to read. And I think this is an area where interspecies comparisons are particularly valuable, because it tells us that the networks around pluripotency and differentiation can form in a multitude of ways. And so comparisons of these will help us identify those features which are really critical through evolutionary pressure for maintenance of pluripotency, and those which are expendable or can be swapped out, and that's hugely exciting.
Anyone else?
I guess my perspective is that I would argue that reprogramming, as essentially defined in an artificial model by Yamanaka, also occurs in the embryo, in the germline. And so I think you need to cast the net wide enough across enough different mechanisms to see the true value of functional understanding. So we can obviously get a good understanding of reprogramming in germ cells, which every generation undergo this process of epigenetic reprogramming. That will complement what happens in stem cells indeed. Some of Sarani's work early was focused on using germ cells as a model of reprogramming, as well as using pre-implantation embryo stem cells to look at the same processes.
I'll just add one additional comment. I think it reflects the fact that in recent years we've seen the emergence of a huge number of databases where we're looking at transcriptional profiling and epigenetic marks in a wide range of tissue types and cell types. We can think of the ENCODE project, the NIH Epigenome Roadmap project, the Phantom project. I mean, the list goes on. And I think that these present an incredible opportunity in terms of being able to find mechanisms that are common if we take a wide range of cells and tissue types, trying to understand common mechanisms of biological function versus cell type specific function, and having perspectives from a wide range of diverse cell types and tissue types, disease types, etc., gives us really an incredible opportunity at this point to be able to make some great headway. And so I think that the diversity is a great opportunity at this point, and I think we've seen the emergence of a large number of data sets out there that are enabling us to be able to study this more effectively.
But, adding on to that, whenever we, in the Phenom project, whenever we add in extra cells, particularly transitioning cells, we find new things. And we're really good at capturing the molecules which are abundant in steady state. And we're only really just now starting to understand the molecules which are transient in rare cell types. But these are probably the molecules which are really critical for understanding how a cell changes to become something else, a lineage specification. So there's a lot more that needs to be done.
Nothing? All right, I'll go. Oh, yep, go on at the front.
With the pig ESLs, do they have, what sort of morphology, the colony morphology of those? Are they rounded? Are they flat?
No, they're flat, but my ESL physiologist explains that because what you're alluding to is the mouse are domed. Naive cell mouse ESLs that form dome colonies. But if you look at the embryogenesis between the two, they're different. And this is where it gets a little bit confusing, but basically he's saying that the way gastrulation occurs in the mouse is different to the pig, and that's how he explains that the two colony morphologies are different.
And have you thought about looking at taking human ESLs and exposing them to the naive reprogramming cocktail and seeing how they compare to your pig ESLs?
We'd like to do all that and more, but we haven't done that yet. We'd actually like to go and isolate, or look at this cell type in humans, see if we can isolate the same cell type in humans. So that's one of the one of the nearer aims, I think, for the work is to go and isolate that cell type in humans and then compare it with the naive and the prime cell types that are out there.
I have a question for Patrick. For those mice that were underweight at birth, do they actually catch up later in life?
So from our work, we don't actually know yet, at least in the enhancer as S2 model. We do know that for the EED weight difference, that is maintained actually for quite some time. From the study of Sarani early on, the enhancer as S2 mice do catch up by about 30 days of age, and so that looks like a true fetal growth phenotype that's then resolved to some extent during the early stages of life. But we know from human studies also, or human patients who are born underweight or overweight, that this can also lead to substantial issues later in life and susceptibility to particular metabolic differences, etc., through life.
With the availability of cosmetic surgeons, there's no shortage of the public going to these people, paying a small fortune, and these people are obviously quite desperate. We've been talking about stem cells as the panacea for all sorts of problems for a heck of a long time. We now have these wonderful molecular tools available. Are we actually any closer, really, to delivering really good outcomes for people?
Well, I'll take a stab at that because it is extraordinarily complicated issue at hand. I think that you articulate that extremely well, and I think that there's it depends on I think if you take into account the efforts in the adult stem cell field, like the mesenchymal stromal cells and all of the efforts that have gone into trying to treat various diseases using those intravenous injections of MSCs, and to some of the more recent efforts in trying to use embryonic stem cell derivatives to try and regenerate different types of tissues or as a model for being able to treat diseases. And so I'm not sure if we're closer per se. I'm not sure that I'm going to get on that bandwagon to say that we're very close. I've certainly been on teams of people that have been working toward regenerating the heart and putting cardiomyocytes that we derive in the lab right into the heart or the myocardial wall to help remuscularize the tissue. And certainly those kinds of efforts are moving forward into clinical trials. But again, when even you say clinical trials, we're still talking efficacy, safety issues, and it goes on and on years and years down the line. And so I'm going to say that, it's there's still something to be realized, I think, in the world of stem cell biology and regenerative therapies, and it's going to be a long road. So I think that my message to the public is just, I hope that this, one of us anyway, was talking about this, the issue of making sure that we get the questions right the first time and getting some knowledge about the basis of these therapies, not jumping in too quickly, which has happened in many cases when you talk about gene therapy or cell therapies. People jump in very quickly. And taking the time to get the biology right. And I think that we talk about these kind of therapeutic panaceas from the very start, and so then the public gets very excited about it with the expectation to be proximal, but that's not going to happen. So I'm not going to put a timeline on it, but I'm hopeful. So that's what I would say.
I mean, I could add to that too, that it's really the problem with these unproven cell therapies. And in Australia, there's been a recent study published, there's 19 clinics operating with unproven cell therapies. Someone died last year, and through negligence, really. And in the reproduction field, out of those 19, one of them is treating infertility. And I recently heard at a conference that there's another person, I don't think it would have been counted in that, well, I don't know, but treating women for vaginal atrophy with liposuction fat. And the problem with this sort of thing is that if something goes really wrong, it sets the field right back. And it needs all the kind of studies at all the different levels that we've heard today, from the very, very basic understanding of what's going on in the cells and before we go into the clinic. It really does need careful work. Otherwise, like the gene therapy got set back when something went wrong. So, you know, it's a difficult area because the media really hype it up quite considerably. And there's people really ready to exploit that, not really caring about the patients. I mean, the clinician I heard speaking, just treating women, when he got questioned about what kind of selection criteria, dose, what kind of clinical assessments and other assessments were done, he didn't have any answer. So women are being treated, in this case, that's sort of an area that I know about, for things by clinicians using cells like fat when they really don't have any idea. They're not gynecologists, for example. They're cosmetic surgeons treating all these sorts of things. They do not have the basic background in that area.
We have to draw a very strong line between clinical trial for stem cell treatments which have got a very solid scientific basis behind them, and where those clinical trials are followed and reported, and not just reported in the short term, but where there are long-term follow-ups afterwards. There are a large number of operators who do not have any kind of clinical trial backing what they do, or they have superficial phase 1 trials which are used as marketing tools. And we must not give these people the benefit of the doubt when we say stem cell therapy. They are not using stem cell therapies, they are using unproven treatments, and they are adopting the term stem cell and misappropriating it. And that's a very important point that we as scientists must state very firmly publicly.
I also just want to make a point, a shout out to the International Society for Heart Research, which has been one of the leading societies to make publicly available resources for people to actually identify these kinds of clinics that are doing cell therapies without any scientific basis, and trying to increase the public awareness about them, because I think they are misleading. So if people are interested, I think the ISSCR has really excellent public resources to be able to understand this a little bit more.
That's why these forums are important, though, because we have people now that are participating in stem cell tourism, going overseas. And there was a paper, I think, in the New England Journal of Medicine about somebody that got a mixture of 3 cell types, one including embryonic, and ended up with a large tumour as a result. So it's important that the public understands where this research is at. So this is why this session is important.
So then perhaps to follow on from that, we are being encouraged to push the translation boundaries in terms of grant writing, and the media encourages us to sell every paper and every story with a headline about how we're curing infertility, or in your case, changing the world of stem cell therapy. What can we do as scientists to help the media get the right information? And as experts in the field, what would you encourage us to do, the rest of us who are not necessarily stem cell experts, but what would you encourage us to do to get the right messages, continue to get the right messages out there, so that people don't continue to pay for these kinds of treatments, which we know are potentially very dangerous and not efficacious?
I think one of the things is that we really have to educate the people to get resources so that they know what to go and ask their doctors, for example. And Nathan has mentioned ISSCR have a very good website and a very good resource. There's resources available in Australia on the Stem Cells Australia net website, and also the National Stem Cell Foundation of Australia as well. But if the thing is, if you're paying for a clinical trial, if they're saying it's a clinical trial, which is what they do at times.
Yeah.
If you're paying, you don't pay for a clinical trial, okay? And if there's no follow-up, and it's really even the assessment of the patient before they even receive the treatment, if all those things are not met, people should not take those treatments. at all.
I mean, the difficulty here, of course, is that these are cells we know very little about, and clearly there's a large amount of basic research which is needed, and so I think that we have to be educating our funders that translation needs basic research, and that's something that I'm probably preaching to the choir here, right? But also, our young stem cell researchers need to be aligning themselves with clinical need, because there are clinical gaps that can be addressed with stem cell therapies if we can get the clinicians and the stem cell researchers speaking together. And then the third aspect, I think, is it's all very well that we stand up and provide information to the general public, but, you know, there's a huge demand for therapies for where there are no good clinical approaches at the moment. And people with chronic pain or who have got degenerative diseases or who are paralysed are really very impatient, and understandably so. So I think it's equally important that we have a two-way conversation with patient communities to understand why they're seeking these therapies, and rather than just us telling them why it's bad, actually learning from those patients about why they're going.
You have a question. Don't know where the mic went though. Oh, there we go.
Is stem cell therapy regulated by the TGA?
That's part of the problem.
No.
I take it it's not.
So autologous stem cell therapy, if the cells are not expanded and treated in any way, gets through a TGA loophole because you're taking the patient's own cells and putting them back.
And not even the fact that you might be processing it in the back, in the kitchen over the stove matters.
These are uncharacterised. This is uncharacterised material going back, which is why I emphasise they are misappropriating the term stem cell treatment.
I realise that,
But if you are doing allogeneic therapy or you are producing a stem cell product, and we have some very good bona fide stem cell pharmaceutical biotech companies here, and some genuine people doing clinical trial, those people are regulated by TGA.
well, is it not worth asking the TGA that autologous stem cell therapy, the regulations have changed?
Yes, and I think that we could all add our voice to that. That's an ongoing conversation with TGA.
I think that another critical issue is actually the way that science is understood in the public domain, and particularly at the political level. I think in Australia recently we've gone through a time, and probably presently, where politicians are not understanding the way science is done and what scientific evidence means. And this is then trickles down to the public. I think it's critically important for science in general that we get the message across about how we do the work and what the evidence means, not necessarily on a specific topic, but generally speaking, that we build blocks, we perform basic research to understand on an evidence basis to make progress. Just like building a car engine, for example. We know you can't make a car go without a particular process to build that engine. We should know that you can't make stem cells work without that basic understanding of how they work, how they integrate into the body, and the other cells that they interact with. And if politicians, or we can get the message across to politicians to promote this level of understanding, then I think we'll be both better off from a funding point of view, but also from a basic understanding of how to go forward and what constitutes good evidence and what constitutes bad evidence.
All right, so with that, I think we will thank all of the speakers today. I think we're very lucky. Oh, sorry, go for it. I was going to go on to say that I think, we're very lucky that the panel here today, that the research field in stem cell biology here in Australia is very bright. So, perhaps it is alarming that there are so many, non-legitimate clinics starting up here in Australia, and per capita it doesn't look very good for us, but I think that the future is really bright, you know, considering the types of research that's being done in this field. So please join with me in thanking all the speakers one more time for a fantastic session.